everyone. Good afternoon, and welcome to the Theoretical and Computational Biophysics seminar this week. It's my great honor to introduce our guest this week, Professor Peter Wolynes from Rice University, from multiple departments. I know that Peter and his groundbreaking work is known to all of us, but by way of introduction, Peter is actually a native of Illinois, Chicago area, and he received his initial education in chemistry, and then he did a PhD in chemical physics at Harvard, followed by a couple of postdocs at MIT, and then he stayed there after a short time.
One postdoc.
For
6 months. So.
Very short. And then actually he stayed at Harvard as a faculty member for a short tenure before actually joining our university at this campus. And he stayed almost 20 years on this campus, during which he did really groundbreaking work in protein folding area, became a National Academy member during that period and went through the ranks, and of course received named professorships. Then after 20 years at UIUC, we lost him to San Diego. I guess this is the ocean factor, I hope.
Yes, really.
And then actually Peter sort of joins chemistry and physics and biochemistry, other named professorships there, and stayed there for about 10 years before he moved to Rice University in 2010-11, where he is affiliated with multiple departments: chemistry, physics, biochemistry, cell biology, material science, and nanoengineering. And he's also co-director of the famous Theoretical Biological Physics Center, where we know a lot of colleagues actually got affiliated with it. So in terms of, I mean, he has a very long list of honors and awards. If I want to list them all, so.
I need the extra 5 minutes for my talk.
Yes, exactly, exactly. But just let me actually just confine myself to mention that he's a member, of course, of the National Academy of Science since 1991. He's a fellow of American AAAS, American Academy of Arts and Sciences. And he's also, just to make the point, he's also affiliated with several National Academy of Sciences in other countries, including German National Academy of Science. He has an honorary doctorate from the University of Stockholm and a Linnet professorship at the University of Cambridge. He's affiliated with the Royal Society and also a foreign fellow of the Indian National Academy of Science. So in terms of research, you are all familiar with actually Peter's work in protein folding, but since about 10 years ago, he also got interested in proteins in brain, which is the subject of what you're going to hear this week. And they have already made actually the idea of energy landscape in proteins and how it can apply also to system peptide aggregation in the brain and its involvement in diseases and health of humans. It's really a great honor to have you here, and we look forward very much.
Okay, it's very good. Well, thank you. It feels very special to me to be invited to give this particular talk at the, I guess, TCBG, which I guess stands for Theoretical and Computational Biophysics Group, which is basically the group founded by Klaus Schulten. I think this is the first time I've given a talk to this group since Klaus passed away. So I'd sort of like to, of course, say a few things about my interaction with Klaus. Klaus and Zan were senior graduate students at Harvard when I was a more junior graduate student. They didn't do a very good job mentoring me, but that's okay. But they were certainly, fun and so on. And then after I went to Harvard as an assistant professor, I also spent a summer in Germany with them and wrote a paper with Klaus that I was just telling people sort of had a second life and is now a 350 citation paper, although it was laid dormant for about 20 years on magnetic field effects. And I had continued contact with Klaus and Zan, usually when they heard some story about somebody in America who was insulting to a German scientist, they said, oh, that must have been Peter. And so I had some contact with them. And then, but off and on. And then one year I got a Christmas card from Klaus and Zan. And that was unusual. I mean, we don't always send Christmas cards to people. We're very bad at that. Got a Christmas card from them, and it was sort of an unusual Christmas card because it also had in it Klaus's CV, which is not your normal Christmas message. But I thought there was something that he was trying to tell me. So here at Illinois, they had started to have some new professorial chairs connected with the NCSA. And I said, well, we should try to hire this guy. And so it turned out to be a little more tricky to get them to come, but they did indeed come here. And I guess Klaus, adapted very well to the environment. So what's interesting is then, of course, when the Beckman started, I was trying to look up the data on this. Your PR has mutated over the years, but the original thing of the Beckman Institute was supposed to be about the brain. And it was supposed to be about intelligence in the brain. And this is what all went on in the PR of what was supposed to go on here. And what's interesting is, of course, actually, this is an example of something honored in the breach. There was no very good neurobiology group here. There was a group of Bill Greeno, but I repeat myself. And in general, there was no brain group. But Klaus was about the only person who did anything connected with neural networks and the brain in those early days. So it turned out I'm a little more guilty person. So I said, well, I better try to learn something about this brain stuff. And indeed, so I could do something so that I could be somehow connected with the Beckman Institute and its focus. And I never did anything on the brain in those days, but I did learn about neural networks, which turned out to be quite important for thinking about folding. And I'll review that in a little bit. But what I'm going to talk about is something about in the last 5 years, I have started to worry about problems in the brain. None of this is going to be something that I would say is, quite revolutionary to the neurobiologists. But instead, I would say what I've been discovering is in thinking about problems that are connected to issues in neurobiology, one ends up thinking about issues about how proteins behave and how they do their business, which has led to many interesting new phenomena, I would say. And that's really what this talk is about. It should probably be called Protein Dynamics Inspired by the Brain. Although I think some of the things I'll have to say are indeed related to neurobiological questions. But when you start to think about neurobiology in the large, there's a huge amount of systems biology that goes on. And I guess the surprising thing to me is that in addition to the complexity of the systems biological level, almost every step that's involved involves new physical processes that I would say the systems biologists usually don't even think they've never heard of or thought about, and even we in protein dynamics haven't thought about. So in a way, the kind of problem, the sort of OR problem of brains is, how do we learn things, and what does it even mean to learn things? And sometimes we have a clear answer. To learn your math, your multiplication tables, the impact of that is you're able to regurgitate mathematical multiplications fairly early. You might learn even a set of algorithms for how to bring problems apart. But of course, in the real world, that isn't, people did not evolve to do that. They evolved to, react to stuff, get out of the way of things and stuff like that. There's actually fairly simple kind of things that all together end up giving complex behavior. I guess the person who really sort of made this clear in the 19th century, someone was talking about philosophy, was William James, who really explains all of our consciousness and everything out of the associations of different memories. Now here, this is a quote from the famous In Search of Lost Time by Marcel Proust. And it's really the clear example of what is association. He says, no sooner had the warm liquid, that is tea, mixed with crumbs, touched my palate. So there's a bunch of physical thing here. It's sensed in some way by the palate. Then a shudder ran through me, and I stopped. An exquisite pleasure had invaded my senses, something isolated, detached, with no suggestion of its origin. And once the vicissitudes of life had become indifferent to me, its disasters innocuous, it's barely illusory. I wonder what kind of tea that was, by the way. But sounds like good stuff. Anyway, where did this come from? And then he realized, and suddenly the memory revealed itself. The taste was that of a little piece of Madeleine, which on Sunday mornings at CaMKII, because on those mornings I did not go out before Mass, when I went to say good morning to my, I think it's his aunt, in her bedroom, my Aunt Leonie used to give me first some of this tea and a Madeleine, and that's how it came about. So there's a real physical memory of something that happened in the past that's sort of being brought back, sort of presumably making the same neural networks activated. And that's sort of the essence of, I would say, learning something. And the only thing that's different about learning, things in our normal world is it's made up of, hundreds of thousands or millions of such events of this type. That certain neural firings at one place cause another set of neural firings to go on afterwards. Now, the key to this was actually something that I did learn about in those early days of the Beckman, that people had thought about trying to model and understand how memories are formed. And this comes after William James and after Proust. There's a guy named Hebb. It's about 30 or 40 years later, I guess. And people have found what is it that causes neurons which fire in some pattern at one time to be have a tendency to do the same thing at a later time. Now you can see I'm using a lot of cagey words there, and that actually means that in fact we don't really understand this. And exactly what are the things that are the same is not presently clear, although it's clear that they're what some of the actors are. So one of the actors seems to be this, that when we have a neuron, neurons have all these little spiky things coming out that are called dendrites, and at the very end they have little nubs that are called dendritic spines. And when electrical signals go across here, usually transmitted by, say, or a neurochemical signal goes across here, calcium is somehow released. In the release of calcium, something goes on in here, and that causes the little dendritic spine to, let's see if it goes, or whether I have to do it. Let's see whether, maybe I don't have the movie version. What you would see in the movie version is that the spine actually grows a little bit. So this is actually and then because it grows, the idea would be, according to Hebb, that because this interaction occurred through some signal that occurred in the past, now this signal will respond even when there's a weaker aspect of it encountered in the future. And so something that happened and worked in the past leads to the same interaction over again. And that's called associative memory. And actually the mathematics of associative memory was beginning to be developed in the context of neural networks by John Hopfield, roughly at the time of the founding of Beckman. So I came across that. In fact, Hopfield's work is based on the work of Little, who was a superconductivity guy, maybe 10 years, say in the early 60s. But it was kind of a low-level thing. Now, in fact, at that time, I couldn't think of how to apply this to anything the brain. I wasn't even all that. I just, even though I'm supposed to work on the brain so that I can justify having an office in the Beckman, I did sort of realize that this could provide an answer for part of the problem of protein folding. And I would say now in the sort of modern way of looking at this, we would say that what we've learned is that evolution is a learning process. And what has happened is in the process of evolution, we may have started with sequences that had all kinds of interactions, say hydrophobic side chains interacting with hydrophobic side chains. But there was no particular, at least this is a little bit of a folkloric thing, but at least we can imagine that there was a situation where we had chains that were basically random in sequence. And in that sort of landscape, you'd have all types of ways of partially satisfying those interactions. You might make some particular hydrophobic guys come together, but because they're connected to some hydrophilic guy, the hydrophilic guys come together too, which isn't good. So we have what's called a frustrated landscape. There's where there would be no particularly well favored state. And then and so this sort of molecule would exist in a series of conformations that are deep traps. They would sort of slowly interconvert and might not be able to do any very specific thing. What's apparently happened to proteins, as Joe Briggelson and I noticed, was that instead, some of these interactions have become strengthened in such a way that a few states, or maybe a small number of very similarly structured states, have all the strong interactions coming together and leave the weak interactions aside without any conflict. So the energy landscape becomes what is now called a funnel, to use the language of Jose Onacek, and the landscape is minimally frustrated. So in this way, we sort of see a connection between the protein folding problem and evolution and learning. Now, this idea we actually turned into a machine learning algorithm very early, and I'll show you in a second. The idea is if you have an energy function that has some ingredients that you might know and understand, you could use the fact that there are known proteins that fold, and you could use those to learn from them what are the kind of parameters in those models that are like the interactions in that model I said before that would lead to funnel-like landscapes. So this is exactly like learning what are the interactions between neurons that lead to reproducible patterns of behavior. And the actual theory worked out with Joe says that the thing that would determine whether you have a fairly unique answer or not is a comparison of a quantity that depends on the energy of the stably folded proteins versus the random proteins. And that gets compared to the ranges of energy you have of completely misfolded proteins. This is something in modern language that we didn't use these words in those days. This is something that we call a loss function in defining a neural network in machine learning. And so we said, well, with a bunch of parameters, let's optimize them to make it give the most funnel landscape possible. And I'm worried.
There.
Oh. Should.
It was folding.
It was folded.
Yep.
Why does somehow the connection to the I'm sorry. This normally has a cursor show up, but I don't see any. Oh, there we go. Okay. So you can see that this is actually not the first result we got in trying to use machine learning, but this is sort of a result from, let's say, I think it's about 2007, something like that, that in 2007, one could learn the interactions in proteins using this sort of neural network Hebbian sort of rule type thing in such a way that you could end up with an energy function that actually allows you to predict structures of proteins. And like I said, this is a result from around 2007. Now, in those days, we were mostly interested just in getting, sometimes folding a protein was an interesting idea. What I want to tell you is that basically over the last decade or so, this kind of thing has become highly reliable, so that the frontier is not so much learning what's going on in folding, we know a lot about that, but using that to understand other aspects of protein dynamics. Of course, this machine learning, so I call this machine learning since 1989. We officially did this with an interaction that was exactly based on the neural network associative memory idea. That was done with Mark Friedrichs. And then later, the optimization based on the loss function was done with Zan and Richard Goldstein, which he published in '92. And of course, actually, why this is not modern machine learning is humans had to learn from this. So we would apply this algorithm and keep refining energy functions to where it finally worked. And so this actually leads to a model that we've jokingly called, one of my students, Nick Schaefer, who's a really great guy, he jokingly said he came up with some acronym that made it sound like it was an awesome algorithm. This was about 10 years ago. And he just was doing that as a joke. I said, no, we'll use that. So the final term is an associative memory, because there's an interaction that depends on things like evolutionary guidance from a database, water-mediated structure and energy model, and it was optimized for energy landscape theory, so it's called AWSEM. And this is the algorithm that results. It's an energy function. So the energy function here and all these interactions are the analog of those connections between neurons that we have in ordinary Hebbian learning. We have to see which ones and which kind should be made stronger than others and which ones should be made weaker. And by the way, in the Cas13, which is not the last one that you've heard about, which was the first one where AlphaFold ranked first. We didn't do that all wonderfully, but we were ranked somewhere like 6th or so. But there are actually several places where AWSEM got better predictions than AlphaFold, but only a couple. They generally beat us. But you can see that as of even 4 years ago, it was kind of routine to get structures in the 1 to 2 to 3 angstrom range. And like I say, 15 years ago, that was something that we could do for a few proteins with reliability. So, and one of the things that's relevant to that and relevant to today's story is that because those interactions are based still fundamentally on physics, even though they're learned with this machine learning strategy, you can also do problems other than just folding individual molecules. You could see how molecules associate with each other. So these are cases where you actually basically know the structure of the protein itself, and then you can use this awesome code to figure out where things are going to bind. And it's a very good binding predictor. This is, again, pretty old stuff, about 8 years ago. So, as I said, starting quite a while ago, we said we understand how to do structure predictions. Let's start using it to understand mechanisms in protein folding and mechanisms in protein dynamics, and also general mechanisms. And in fact, there's a problem that I started with a student named Ming-Chen Chen. I'll come back to that problem in a few minutes, but the problem had to do with a protein that's involved in long-term memory. And so we started to use the AWSEM software to predict and study proteins that are involved in various neurobiological stories. Now I'm going to deviate from the historical record to start at more the beginning of the process, because that particular protein which we studied, I'll come to in a moment. I'm going to talk about another part of the memory process, that first part of how does a dendritic spine change its shape. And this was actually work that came about partly because my colleague Margaret Chung, my then colleague Margaret Chung, she's at the University of Houston, but she's part of our center. She was already interested in the proteins that were involved in neural regulation. And now that AWSEM was in good shape, we could start to look much more deeply at the proteins that were involved in the process of remodeling the cytoskeleton in response to electrical signals. So here's a picture of that again. We have a small dendritic spine. When neurochemical signals come in and cause eventually calcium to be released. And the calcium release seems to have the effect of changing this pre-existing actin cytoskeleton to have a different structure. And it's a structure in such a way that the actin cytoskeleton expands. It actually expands a little bit, then it contracts, but it remains slightly larger than it started with. And then it apparently can stay that way for very long periods of time. Very long periods of time in in most of neurobiology, in this part of the problem are, minutes to hours. If you try to keep a mouse alive for 2 or 3 weeks, that costs you money because you have to pay for them in the vivarium. It costs the same as putting your family up in a hotel. So you got to make sure they don't live to be 50-year-old mice. So this doesn't deal with the problem of very long-term memory. It's only the problem of sort of short-term memory. And the first part then will do the sort of, it's called long-term potentiation, but it's actually relatively short memory compared to, remembering what your aunt made for cookies. Okay. So it turned out that one of the proteins that's key in this is a protein called Calmodulin dependent kinase 2. And this is, as it says, it's an enzyme, but it's an enzyme that's present in such large concentrations. It's actually present in a concentration almost not too much smaller than the concentration of the actin, maybe down by a factor of 10. And it was clear by some pictures, which I'll show you, that actually this enzyme forms structures with the actin, and it's partly this release of an existing structure by the calmodulin as part of the calmodulin dependence, and the reconstruction of a new cytoskeleton after the end of that. That is at least part of the story of how you get this first remodeling of the cytoskeleton, the expansion of the dendritic spine that's being signaled to. And one of the points about this is it's both relatively stable at the end of all this, but it is also regulatable. You could do this again, make it bigger, bigger. You also have ways, perhaps, of making it forget things. Actually, one of the consequences about fooling around with Calmodulin kinase is people at least report some drugs that have the effect of making it hard to remember for a long period of time something that was frightening. So this is something people think about as a treatment for post-traumatic stress disorder. But of course, it's pure witch doctory how it's used so far. As you see, the system biology of this is very complicated. There's actin remodeling that involves all these different proteins, and then in addition we have all these factors afterwards, and then we have this one little thing of calmodulin kinase and calmodulin. So our exercise was, since we have structural prediction tools, was to try to figure out how does calmodulin kinase interact with actin, and can we predict why it would, and can we understand why calmodulin, which is what comes in when the calcium signaling starts, why it would allow this new structure to reform. So it turns out, even though we learn things and can predict structures of protein molecules, we can do that better the smaller the molecule is. And of course, if we already know the structure, you're worried about this one?
The battery is kind of.
Is it? I just changed the battery.
Yeah. Oh, okay.
It can't be the battery. I think I'm going to use this one. I got this from Jim Simons. So
okay.
it's
maybe
nicer.
it's context.
It's nicer. It's richer. Although if he say Beckman, maybe I should steal that. Okay. But anyway, so actually, parts of the structure were already known. So for example, the kinase domain, the part that does enzymology, actually was known by John Currier, done in a standard X-ray crystallography way. And then the thing actually forms this kind of hexagonal nut. And so it's actually present in the form of a sort of Christmas ornament-like oligomer. And on the other hand, there are big parts of the protein that no one knew any structure of. There's a thing called the association domain. There's a thing called the linker, and there's also something called the regulatory domain. Now, these things were all learned that there were these parts that changed the overall binding and activity of actin by very difficult cell biological and neurobiological experiments. So I can't really treat fairly all the things that explain why they have these different domains. But our job was then to try to see what the function of this remainder of the molecule was. And we were helped in some ways. Ming-Chen Chen noticed that when there are actually lots of structures of proteins binding with actin, and in all of them, there was kind of a common pattern of the general area in which they bind to actin. They have a sort of common hydrophobic cleft. So we could kind of make a guess on this molecule of what part of it might be the part that sort of fits into the actin. So we were able to first do that to make a more precise prediction, starting by knowing a structure for F-actin. That's pretty big. We have our CAM kinase monomer. We know its structure by itself. Then we can put a sort of general force sort of pushing you in the general neighborhood of where we would think a binding site would be. And then we let the awesome forces that include these water-mediated interactions that hold together many complexes. And we then led to a prediction of a structure. This was work with Margaret Chan Wang, who I think he's often a postdoc at MIT. He may be finishing that up, and Ming Chen and myself. And actually, a structure was predicted from this. And the main thing that you get out of the structure is what's this sort of orientation of this, exactly where does this bind? And the main sort of low resolution thing you get out is what's the angle at which it fits into this cleft. And then this could be compared to cryo-EM structures that we determined by Neil Waxham, who's one of our collaborators, and turned out the predicted structure and the cryo-EM match up quite well. One of the things that then follows as a consequence of this is actually, say human intelligence, mostly the Chan's human intelligence. Once you know the direction of that binding site and you know what this angle is, you can make predictions of what larger complexes would look like. And the main thing is that actin itself is a helix, so it has a certain periodicity. And one can find out that if you really want these to fall into more or less periodic structures, they have to be 36 nanometers apart. And that's one of the predictions in the model. And you are then able to make bundles of actin where the actin is held together by these little nuts of Calmodulin kinase. And you could form bundles, but you can also form a structure where things meet at an angle. And you can form other structures of this type. So it turns out that the structures that you predict by applying periodicity plus the known docking and the known angle tell you about how these bundles form. And then also because of this offset of the angle, you find that in general, you can only do this for a finite length of actin. And after that, they really have to come out of phase. And so this actually turned out to determine more or less the length of bundles that we're seeing in the electron microscope. So, okay, so now we know that, the catalytic domain, whose structure we know, we can dock it, we can figure out what structures we get, but what is going on with the, the parts that had no structures, the parts that were sort of intrinsically disordered, if you will. Well, Waxham, he did an experiment in which we cut apart this molecule into little short peptide segments, or you cut it apart mentally. You then pay $7,000 and you get an array which has small short peptide fragments on here, and then you pour actin on it and you see where the actin binds. And then this gives the pattern of actin binding. So this gives you a presumed idea of parts here that are likely, but they're certainly not certain, likely to be the parts that are going to bind to the actin. Now, we actually did this with another protein that I would come up with later, where this was work that Nick Schaefer did in my group, and we couldn't get Neil to pay the money for the corresponding slide here. So we paid the money for that other protein. So I think by the definition of what's an experimentalist, I became an experimentalist because I paid the money to do the experiment, right? But actually they still did it in their own way. But this he paid the money for, so he's the experiment. So we have an idea of what parts are more likely to bind, which ones are less likely to bind. And that, again, can be a kind of soft guide for the binding process. So what we did was we first take one of our, we just took this sort of disordered part, sort of generally pushed it in the general direction of those binding sites, but then let AWSEM take over and let it run without any bias to any specific region, and then see what structures result. And we end up with a structure that looks kind of like this. So this is an interesting thing because the geometrical complexity comes from this very nice, sort of very rigid object, more or less doing classical folded protein recognition. But then these other parts that are intrinsically disordered actually do fall and live in distinct places on the actin. Now, how then does this work? This gives sort of geometrical specificity, but this binding, because it's at least connected, gives you an oomph. It gives you an extra total binding free energy. And that's what allows these regions to be able to act as regulatory regions and to determine whether you actually can associate. So all the domains of the CAM kinase are used to bind, not just the one that was there. And it's the regulation of this sort of secondary binding that is involved in how Calmodulin binds. Now, one of the things that we can also do with AWSEM dynamics is we can do things like calculate free energy costs of pulling parts of the molecule apart. What one discovers is that the regulatory domain and the linking domain and the association domain are all pretty much independent in their binding characteristics. You can pull one apart and not the other, and so on. Together, then you might have a total binding constant of binding free energy is quite large, but it's made of 1.8 kilocalories coming from here, 3.8 from here, 3.7 from there. Those all add up to a large and strong binding, but each one is actually fairly modest and weak. And that turns out to be, so we have, say, a 9 kilocalorie binding. So that's why this thing, once it's finally bound, is quite stable. But you can see that each part of it is going to be fluctuating on and off. And that turns out to be the mechanism of action of Calmodulin. Calmodulin is activated when it has calcium in it. It's a protein that binds calcium and changes its structure, which also is quite flexible. And what we saw was that when we looked at how Calmodulin binds to CAM kinase, we see that it actually competes for the same sites, that we have to use for the binding of actin in these regulatory domains. So what we have is an example of what in another context we termed that Betsy Komoves coined. We have a phenomenon of molecular stripping. At equilibrium, this is a very nice folded bound molecule, but when I throw in Calmodulin, Calmodulin comes in, rips off one part of the molecule because that's fluctuating on and off, and then once it rips it off, then the whole binding constant has gone down to a much more modest level, and that's why you're now able to rearrange the Calmodulin. So this is how you get something that's very stable and very strong until Calmodulin comes in, and then by molecular stripping, the Calmodulin allows this thing to fall apart, and then it can come together in new structures, which presumably there's other features that are allowing it to expand. So now I'll turn to, I guess I've been talking a little bit too slowly. I know I'm going to go over time, so I apologize to the Zoom people, and to you too. But actually, the protein the protein that got me into doing memory stuff was not the Calmodulin, but was a protein called CPEP. And the reason is this protein was postulated, and there's evidence for the fact that this protein was responsible for the very longest time memories that we have in neural systems. In fact, this problem that there's a difficulty with very long-term memory was identified by Francis Crick in 1984. He has a variety of mechanisms by which you could have things that last many, many years. The problem is, of course, proteins are always turned over, except in places like the eye, crystallins. They're turned over typically in hours or days. So how can you remember anything about your aunt from 30 years ago? It's got to be, you have to figure out some mechanism to create locally in every dendritic spine something that's going to last for 30 or 40 years. Well, one of the mechanisms that Crick proposed, he proposes several. One of them was, in fact, if you form some kind of aggregate in this region, then an aggregate could be much, much more stable because it has many more interactions. So it could be so stable that it would last for a very long period of time, certainly more than 24 hours, even if the mean half-life of protein, say in yeast, is only 42 minutes. So following up on this idea of Crick, Eric Kandel actually identified with Susan Lindquist a protein that seemed to be forming aggregates in, I think it's the sea slug called Aplysia. These aggregates actually are the infamous amyloids that we have in other kinds of, that we think of as baddies in diseases like Alzheimer's and so on. But here, it's what it seemed to be essential to form these plaques of, sorry, these aggregates of the Aplysia CPEB protein, which is a cytoplasmic polyadenylation element binding protein. And if you say made mice that had damaged CPEB, they could no longer learn anything. I mean, you would throw them into a thing of water where a normal mouse would learn after having been thrown in there that there's a way out of the swimming pool, but the CPEB damaged mice would all drown, So I'm exaggerating the statistics. More of them would drown, and so on. Murdering mice is very time-consuming and expensive. Okay, but Aplysia, also nobody cares about Aplysia when you probably could eat Aplysia. So in Aplysia you can do the same thing. Aplysia learns not to that certain chemicals are going to be noxious, so it avoids them in that sort of Pavlovian response. But again, if its CPEB has been modified, it doesn't. So Ming-Chen Chen, this is how I got into it. He was a rotation student, and I give rotation students a protein to look at its structure or predict something about it just for fun to see if there's something there. And I gave him this protein, and he predicted that it was a helical bundle that formed dimers, trimers, and tetramers. Well, in fact, some of the evidence was that normally CPEB, when it's not in that aggregate, really is in a helical state like this. And Ming Chen noticed, however, something that is kind of obvious, but I didn't think of it, and probably many people noticed, a beta strand, an alpha helix is coiled up, a beta strand is much longer for the same number of amino acids. So that means if you pull on an alpha helical element, if you can somehow grab onto its ends and pull it, it will then have a tendency to become a beta strand and can aggregate. And in fact, we then were able to compute how much pulling force you would need. And you don't need a lot. You need the amount of pulling force you can get from actin. And in fact, Kandel had shown that the CPEB3 aggregation requires actin. So you need to pull on the molecule. Then after you pull on it, you're able to cause the aggregation of the CPEB3. Well, how does that do you any good? Okay, well, first of all, you've got some new long-lived structure, but it's also interesting that, although we've said like the A beta amyloid is something that everyone recognizes as being bad, in the case of CPEB, it turns out that it's actually more active or acts in a different way when it's in the aggregate form than when it's in the monomeric form. In the aggregate form, it turns out to be a protein that activates the translation of actin mRNA. But when it's in a monomeric form, it represses the activation of the mRNA for actin. So you can see that when you make this, this fiber, once you've made the fiber, then you're going to have a feedback loop where you're going to make more actin. And so the fact that you need more actin to assemble it will be taken care of. So there's this kind of positive feedback loop that's involved in CPEP. Now, I know I'm going way over, but I do want to tell you about an unpublished thing that I think is really exciting, because one of the questions that occurs to you is, why should the aggregated form activate the processing of mRNAs? That sounds a bit strange. There's got to be something going on in that. And actually, present student in my group, Xinyu Gu, had a really great idea about how that works. And I'm going to show you her idea. And the only thing that I did was provide the right math to do it. But she had the physical idea behind this. So it turns out she'd also noticed that of all the RNA binding protein coding genes in the human genome, 72 of them are binding RNA, and 500 of them bind RNA. 240 proteins have prion-like sequences in them, and 72 are common. So it seems to be a common thing that you form something like this prion or this aggregate out of RNA binding proteins. And it turns out that the structure then of the RNA binding domain of these proteins was known. When they exist in a monomer, they're understood, but of course, part of the molecule is also, again, this sort of intrinsically disordered part that has some recognition sequences. There's another part called a zinc finger. And what was found was there's at least 2 or 3 other proteins where it's known that the aggregation either makes the protein into an activator of translation or a repressor of translation. And it seems to be that there was no particular rule to this, and there are actually only a few cases where this has worked out. In the case of CPEP, the aggregate is an enhancer. In the case of this protein RIM4, it's a repressor of the corresponding DNA. There's another enhancer. And one of the questions is, how can an aggregate sometimes repress and sometimes activate things? And so it turned out that the difference is which part of the DNA, sorry, the RNA, binds to the CPEP. So in some places, you bind the start signal to the CPEP. In the other place, you bind the end signal of the RNA to the CPEP. So that means that you actually have a structure which is something like a core of an aggregate. And then when you put the RNA on here, you have either the start signal or the stop signal all being connected to the aggregate. And that would be the difference between the guys that enhance and the guys that repress. So here it's kind of drawn. So Xinyu had the idea, well, look, there's really 2 very different situations. If you bind here, maybe I inverted it the way I said it. If you bind at the outside, you then a ribosome. The ribosome binds the RNA, transcribes it, ends up in the center. It then has to diffuse out and maybe rebind and keep recycling. The problem with RIM4 is it has to bind at the place that's already connected to the aggregate. So it has to work its way through the aggregate and then goes backwards the other way and then diffuses out and falls off. So this phenomenon of worrying about where stuff goes between different enzymatic events is a phenomenon called channeling. You may have heard about Andy McCammon using this idea in metabolic assemblies. But here, what's interesting about it is one of the steps of this is this processive part of ribosomes moving along the RNA, which has a vectorial character. So we call this vectorial channeling. And we simulate that by writing down equations for the diffusion of ribosomes. Ribosomes they of course both diffuse, and also they get translated vectorially along the RNA sequence. We know the rates of translation of ribosomes along RNA. And so what we have is a situation where there's diffusion processes here coupled to a processive system here. So we can write down a kind of coupled diffusion equation and translation equation. And the only thing that's interesting about it is it depends on the structure of the places and locations of all the different RNAs. And that depends on whether they bind to an aggregate. So we can work this out. We get an interesting electrostatics problem with sources, but you have to solve the problem self-consistently, which this is where I jump through it. Of course, you can use the techniques of Green's functions if you took your first class in mathematical physics as a graduate student. And we can work this through, and without doing any simulations, we can then predict whether in different geometries, how much the enhancement would be as a function of how many guys you aggregate together. And it's actually a little bit stronger if you were to form spherical aggregates than cylindrical aggregates. In case of Rim 4, you do instead this repression phenomenon, that's why everything's blue. And then one of the other phenomena that does at least tune it, you get an effect without this, but if the diffusion coefficient inside is slow, that actually makes the recycling process even slightly better, and that's why you tend to get, if the diffusion in the interior of the aggregate is smaller, you get a big enhancement of this effect. So I think you can put in numbers that at least shows that this kind of model makes some sense, and it'll make predictions about other proteins. Now, I really have gone over the time, so unfortunately I won't tell you about aggregation landscapes that are involved in disease. We've been studying amyloid aggregation. We've been studying the aggregation of a protein called tau, several other cases. One of the proteins that we've studied is Huntington's disease, which turns out to be relatively similar to, has many similarities to the CPEP protein. And it has this weird phenomenon that it involves a polyglutamine sequence. And the time when you get Huntington's disease, if you're going to get it, depends on how long the sequence is. So that if you have a sequence that's say 60 and long, you're going to get it when you're a teenager. If you have a sequence which is 25 and long, like everyone in this room probably, you'll make it to, you'll make it to your ordinary death. And this is the time to get Huntington's disease. I don't like to, the person who discovered this is Nancy Wexler, who's a very, very nice person. And sadly, she got into this field because her family had Huntington's disease. And she's unfortunately, since this picture was taken, when I saw her, she was in pretty good shape like this, but she's since gotten Huntington's disease. So that's sad, but she's the one who discovered this length dependence. I'll just explain that, in fact, again, one can do simulations of this and actually predict why this critical length. And the main thing that is the sort of bottom line is that when you have short sequences, the polyglutamine remains fairly long, but when you have a long enough sequence, it can double back on itself by itself. And that internal extra loss of entropy is what makes the longer sequences more dangerous. It's okay, I'll just step ahead to talk about the people who are at, not CPEB, but the Center for Theoretical Biological Physics. We have a whole group that's studying problems of memory. These are my students. This is Nick Schaefer, who was my student and then postdoc, and now he's a little bit bullheaded, and so he won't leave Houston. So he runs a machine learning for profit company. He's really one of the best students I've ever had. And then Margaret Chung is involved in the early work in AWSEM. And our funders are the center, money from CPP and from the NSF grant, also separately to study dendritic spine. So with that, I'll stop and take questions. I apologize for going over. Thank you.
Thank you very much. The biology continues to amaze us, it seems. I mean, so many complexities, you know.
Well, as I said, every problem that we've looked at, not just in this area, but in chromosome dynamics and gene regulation, every time we look at something that a systems biologist says this inhibits that or this does that, that's true, but we discover new physics and how that happens in ways that people hadn't anticipated, I would say. So we're always surprised.
Transition between helix scalar sheet. So you think it's a kind of reversible process at the biology scale, kind of we go back and forth?
Well, it can be somewhat reversed. And in fact, another thing that we're just finishing up is if you sumoylate, there's a thing called small ubiquitin-like modifying protein or something called sumo. That sumoylated form doesn't aggregate. And so there are ways that you can say, somewhat break up some of these aggregates. The problem is that when you have a very big aggregate, it's just like that issue of the actin Calmodulin kinase binding. Every individual part has a small binding energy, but you're adding up lots of those. And so that's why you get to have something that lasts for 40 years. Prions were discovered originally by people sort of discovering that you could hardly sterilize stuff if you operated on someone who had Kuru or something. And you said, well, I take my my scalpels and run them through my autoclave, and now you use them on patient number 2. Patient number 2 got neurodegenerative disease. So most of the I forget the name of the corresponding disease that's not in Kuru is the disease in the found in the people in New Guinea. It has another name in the West, but basically people were getting this disease because it was a standard test of a neurologist to come to you, and a neurologist would try to see if the nerves in your hand were working. So he would take a pin you with it. Doctors have to be a little bit sadistic, right? So he would pin them, and he would always use the same pin. And then they discovered, well, all this guy's patients would end up having dementia later. If they didn't start with dementia, they end up with dementia. And then they finally figured out this is so stable, you better use new pins every time you pin people. So even very simple parts of physics can help medical practice.
Turn on the lights.
So Peter, you didn't have a chance to talk about it, but tau seems to be sort of an interesting discovery in memory and the action of neurons. What were you guys looking? What aspect?
What have we done on tau?
On tau.
On tau at this point, we've actually just done studies of the nucleation and growth. We have and found a kind of interesting thing that unlike well, first of all, almost every model of aggregation of both A beta and tau is oversimplified. So in the case of A beta, you typically have a tip. And the tip of the thing is, most people think it's just like you took the protein and cut it off. But it turns out that no, there's about at least 3 or 4 molecules as you move in from the tip that have become disordered. So a freshly cut A beta fiber grows faster than a typical A beta fiber. Now, interestingly, in the case of tau, you have sort of 2 different states, one of which is a fast growing and one of which is a slow growing state. And they're kind of in tandem equilibrium. So I think there's a lot of questions about, you might say, the evolution of aggregates.
Restructuring.
So for example, just recent discovery on tau, this is an experimental discovery, of course, is that people with different diseases have different polymorphs of tau. So the polymorph is characteristic of exactly where you have the tau problem in your brain. And so one of the things we're going to work on is how that growth process is different and how it's different in different conditions and so on. So that's about, we haven't done as much on tau as we'd like. And so it strikes me as suspicious that these proteins all have sort of structures that are sort of reminiscent of what happens in the Huntington case and in the CPEP case, but none of the models of neurodegenerative disease focus on them actually somehow interfering with the memories itself. As you know, Chris Dobson and all these people say, oh, it must be a form of oligomer. The oligomer causes some other problem like just kills cells. But it's just suspicious to me that they also look very similar to what goes on in actually positively forming memories. So, you know, that's one of the angles that I want to explore, but we don't have much about it yet.
Shall
I was wondering whether we can actually, by looking at either the structure or the sequence of the oligomers, whether we can predict whether it's going to bind the mRNA at the 3' or 5' end.
Yes, we do that. Yes, yeah, that we've done actually. That's why I told you where we knew where it was. Okay, yeah, I didn't say much about it because I was rushing.
Yeah. So,
Oh, yeah.
yeah, Peter, and based off of our earlier discussion, you mentioned that the water was kind of important in these binding simulations, but are there, what effects are important to think of in these binding simulations that might not be thought of in just folding simulations, that might go into potentials or your prior?
Well, like I said, I think these water-mediated interactions are, well, I mean, how we discovered them is, you know, by humans learning from machine learning. Gary Papoyan, who was in my research group, we had done papers on folding. We had a good sort of okay predictor of things based on contact interactions between amino acids, like the ones we normally talk about in elementary folding theory. And I said, well, why don't we look at known interfaces and see whether they have funneled interfaces or not? And actually, it's an interesting question because you don't have to be as funneled to bind because you don't have such a big entropic problem to overcome. If you have already folded things, the search problem is smaller. It's still significant. So the answer could have been, none of them are funneled, and you just have to try every binding site you want. But what he found was about half of them were funneled, and half of them were not funneled. And so probably again, I would have just said, that's the way it goes. It's just sometimes it's funneled, some of that. But he noticed that the half that were not funneled always had waters found in the interface. And so then we said, well, that must mean there's a special kind of interaction in which a water simultaneously is coordinating with a guy on the left and the guy on the right. And once you've thought of a form of an interaction, you throw it into the machine learning algorithm optimizing TF over TG, and you find out what value it should have. And then that's what actually made the prediction of interfaces come out very nicely, and all the interfaces of proteins that anyway had structures in the PDB had funneled interfaces. Now there may be even still weaker bindings that are not very funneled in their interface, and that's its own interesting problem and is related to what people call fuzzy protein complexes. And Monica Fuchsreiter is kind of, you know, a big advocate of those, and we've actually shown those, some of the things that are fuzzy complexes have, even with the water-mediated interaction, have very weakly funneled and frustrated interfaces.
I have to ask this following up your question. Sorry. So
Yeah, sure.
disordered proteins and ordered proteins must be different in terms of water-mediated interactions.
Well, it's you know, I think there's, let's say the tack we're taking is that basically the interactions are the same, but of course there's a different degree of biasing for in one case than another. There's different entropic amounts that you have to overcome. You know, there's an interesting, I have to be, well, you have to be a little careful about this, but I think that about saying anything about this. people spend a lot of effort trying to come up with potentials that give the right structures for IDPs. And the right means something like, say, radius of gyration, something like that. If you look at most, like the D.E. Shaw potentials with ST, whatever water he used, almost all proteins in their unfolded state are quite a bit more compact. And so people have been fooling around with that. So there's a kind of small industry of taking some experimental interaction and then some experimental measurement on IDP, and then using that to sort of bias your potential. So Bin Zhang has done that, Garrick Popoyan has done that, and that works. Now, one of the things that's tricky here, and it's actually, I think, the main point of the paper I wrote with Garrick about that he invited me to be part of the paper, was I said, well, the problem here is that the definition of a disordered protein is that it has a large core, that is, the fluctuations are large. And if you do perturbation theory in statistical mechanics, you discover the sensitivity to an error in the potential is proportional to how big the fluctuations are. So I think that this is the fundamental reason why, that they're trying to get the actual radius of gyration of unfolded proteins is very, very difficult, because by definition, they're the most sensitive to things you screw up. Now, the flip side of that, though, is the same statistical mechanical theory says it doesn't change anything about equilibrium constants. The change in structure is first order, but the change in free energy is second order. So it seems to me that people who are trying to do that, it's kind of, they're taking on a hard task. And in most of the biology, you saw that it was the binding energies that mattered. So you could have a pretty bad structure and still have a pretty good binding energy, I think, for the IDP. So don't tell that to the section of the Biophysical Society to go to the IDPs.
Yeah.
Probably doesn't matter. They don't.
Hey, Peter.
Yes.
Imagine they're like hybrid of the ring for CPAD, which a protein that already they will organize mRNA opposite direction, alternate each other. Would they like further enhance the efficiency?
I think that'd be an interesting thing to do, to play with this, and see if you can come up with, constructs that would do other more interesting things. You could almost imagine, I mean, this is in my more philosophical mode, you could always imagine this is how this kind of effect in the distant past was how you ended up with the more complicated organelles that we see in cells that involve, several things, coming together, that it was just originally a little bit better doing some channeling, and then it gets better and better if you hold them together more rigidly, and maybe you can start doing more subtle stuff. And so that's how you end up with ribosomes and end up with all these other objects. But that's back to, origin of life stuff, and, that's hard to actually, it's good for writing papers, but hard for convincing other people that you're right. You have to be kind of mean, let's say, but like I'm thinking of some people. You have to be kind of difficult to convince them that you know what you're doing. So I may do that as I get older.
Hi, Peter. I have a question. It's also kind of philosophical. Do you think that in order to form the physical structure for the underlying memory, that depends on the macromolecule conformation? In other words, do we need single genetic spine to form a memory, or do we need a set of spines to coordinate and work together?
Good question is all I can say. I think that's quite an interesting question. It's one that's sort of on our mind, and one of the problems is, you have a lot. I mean, one of the advantages of storing information in the dendritic spine is it's a lot smaller than the whole neuron. So a real neural network could already be made by, let's say, 2 neurons with just the right pattern of the dendritic spine. Now, of course, their electrical signaling, sort of involves more than one spine often. So it might not be that every dendritic spine is actually the ultimate primordial unit of memory. It might be multiple ones, but I think it's a great question. I mean, there's so many things I showed you back the beginning of this systems biology. And you can see that this is just a tiny little part of it that I showed you. There, it's here. Look at that. I mean, look at the number of proteins that are involved. I didn't even list CPEP here. I didn't list all the mRNAs that are being processed. I mean, the actual system biology thing is large. And then, of course, there's also a physics question of this stuff is a kind of a gel, the cytoskeleton, but in a sense, that's kind of a glassy thing. So it depends even on how fast this forms and how slowly it forms. And that gives its own physics questions. So, I mean, that's from my point of view, that's one of the things that's fun about it, namely that it sort of gives rise to so many interesting physics questions. From the point of view of, curing people's diseases, that's maybe distressing, but because it means we're very far from understanding what's going on here. I mean,
Peter, really? Can you hear me?
I hear a disembodied voice.
Yeah.
Oh, who's that?
It's Ralph. Hey, how you doing?
Oh, good. You're so small there. Sorry.
So what I'm curious about is that, like, if you look at the clinical description, frequently it'll be specific proteins as a biomarker with their indicated phosphorylation levels, and it seems like it connects to this slide in a
Yeah, exactly. This Calmodulin kinase is itself a phosphorylating agent, and it even autophosphorylates itself. So it's possible that some element of the memory is stored in the range of phosphorylation states that you have. And it's not all in structural. In fact, there's maybe two schools of thought about Calmodulin, but mostly the memory is stored in phosphorylation states of Calmodulin and other proteins, and others that is stored in the structure that results here. So that's definitely an element of what's going on. In the case of tau, for example, tau aggregation depends a lot on phosphorylation state. So we studied that also, by the way. But so there's all kinds of levels of interesting structure here that we haven't talked about in these things. That's why, like, in a way, I would say this is brain-inspired biological physics, as opposed to telling you why we now have the grand unified theory of the brain. I don't have a grand unified theory of the brain, except probably at the end someone will say, "Oh yes, it was a Hopfield model." But exactly how it became a Hopfield model will be many more years, I think, to figure out. Okay.
Any other questions?
Good to see you, Rob. Yeah.
If not, please join me to thank Peter one more time.